STEMMEDICARE, Noble Care for Human Nature, Extracellular Vesicle Therapeutics for Infectious Diseases based on Primal Immune System. The COVID-19 pandemic has raised one question. Why do children have more asymptomatic cases, while older people have higher infection and mortality rates? What causes the increase in the fatality rate of COVID-19 with age? Finding this answer has led us to take a fundamental look at the existing immune system. The immune system is our body's self-defense mechanism that protects us from toxic substances or external infectious agents. The immune system is generally divided into the innate immune system and the adaptive immune system. The innate immune system acts as the first line of defense that is activated within a few hours after infection, and immune cells such as phagocytes, neutrophils, NK cells, and dendritic cells, as well as natural antibodies and complement proteins, are involved. The adaptive immune system is a learned immune system that is formed several days or weeks after infection, and cytotoxic T cells and antibody-producing B cells play a major role. Antibodies are substances that protect our bodies by binding to antigens, such as toxic substances or foreign infectious agents, and neutralizing them. Antibodies are also generally divided into natural antibodies and adaptive antibodies. They play various roles such as defense against pathogens and maintaining immune homeostasis. Natural antibodies refer to polyreactive antibodies that exist in our bodies from birth, and they play an important role as a first line of defense that responds immediately to all antigens along with complement and activate pathogen-specific adaptive immunity. Representative natural antibodies include IgM and IgG3. Adaptive antibodies are produced by adaptive immunity, especially B cells, after infection. They react only to learned antigens because they have high specificity for specific pathogens. While children produce adaptive antibodies within 2-3 days after vaccination, it takes longer time for the older to produce them, which increases the mortality rate from SARS-CoV-2 in the elderly. So how are fetuses protected from infectious agents during pregnancy when the immune system is not yet fully formed? Fetuses 15 weeks after pregnancy can receive maternal antibodies through umbilical cord and be protected from the external infections. So how does a fetus protect itself from foreign pathogens before 15 weeks of pregnancy? We found for the first time in the world that fetal stem cells in the early stages of pregnancy secrete natural antibodies and complement that can protect themselves from external infections. We were the first to confirm that extracellular vesicles secreted by fetal stem cells early in pregnancy contain natural antibodies such as IgM and IgG3 and various complement proteins. We developed a novel ex vivo culture system that simulates the environment during pregnancy, and we established fetal stem cell lines that secrete natural antibodies using this culture system. The extracellular vesicles secreted by fetal stem cells we have established contain natural antibodies, complement proteins, and interferon-induced proteins that can protect themselves from external infectious agents, as well as HLA-G, which induces immunotolerant environments that protect themselves from the maternal immune system. Specifically, we newly defined natural antibodies secreted by fetal stem cells in early pregnancy as primal immunoglobulin, called IgP, to distinguish it from antibodies secreted by B cells. The primal immunoglobulin refers to a pluripotent antibody secreted by fetal stem cells in the early stages of pregnancy. It has the same structure as a IgG3 antibody among previously known antibodies. When comparing the structural characteristics of IgP with IgG1, the most common antibody, the following differences can be seen. Compared to IgG1, IgP has 11 disulfide bonds in the hinge region, so it can bind to a wider range of antigens. After binding to the antigen, it has better neutralization, complement, and cytotoxicity activation effects than IgG1. The most important difference is that IgG1 can only bind to educated antigens, whereas IgP is a natural antibody that has not been educated to any antigen and thus has a primal repertoire capable of responding to all antigens. We first proposed the concept of the early pregnancy fetal immune system established by extracellular vesicles containing IgP, complement proteins, and interferon-inducible proteins with various antiviral effects, as the primal immune system. The primal immune system consists of three defense mechanisms: extracellular, cell membrane, and intracellular defense mechanisms. The extracellular defense mechanism is primarily driven by IgP and complement proteins. IgP can recognize and combine pathogens that enter the body to neutralize them. In this process, the complement system is activated by IgP bound to the pathogen, and the complement protein binds additionally to activate phagocytosis through opsonization. Pathogens absorbed by phagocytes are broken down, and the broken pieces are delivered as antigens to antigen-presenting cells for activation of pathogen-specific adaptive immunity. In addition, IgP bound to pathogens activates antibody-dependent cell cytotoxicity that removes pathogens by combining with various immune cells such as NK cells, macrophages, dendritic cells, and neutrophils. The second is the cell membrane defense mechanism. Viruses that enter the body bind to specific receptors in the cell membrane and enter the cell. Then, they infect the cell and our body through rapid replication. However, when extracellular vesicles containing IgP are absorbed into the target cell, interferon-inducible proteins in IgP-EV reduce the expression of specific receptors that bind to the virus or block the receptor to prevent the virus from entering the cell. The third is the intracellular defense mechanism. The virus that enters the cell replicates itself inside the cell and secretes it out of the cell, infecting other cells and our entire body. However, when extracellular vesicles containing FARS-GP are absorbed into the target cell, interferon-inducible proteins in IgP-EV combine with viruses that have entered the cell to suppress the replication of the virus. At the same time, autophagy, which breaks down impurities such as viruses inside cells, is activated by IgP-EV, and viruses are broken down in autophagosomes to protect cells from virus replication and infection. IgP-EV can be used as a general-purpose immunostimulator in combination with various vaccine antigens such as inactivated vaccines, protein and peptide vaccines, RNA and DNA vaccines. In addition, it can be used for the prevention and treatment of various infectious diseases by itself, and can also be used in the field of developing therapeutic vaccines such as AIDS and cancer.